to Henderson, which in the 90s was beyond the reach of an eradication project. And people said to me, perfectly reasonably, we accept your data from 91-92 concerning rat-induced mortality of the chicks, but was it typical? So these are the data for Murphy's petrels from 91, and basically what we're seeing is around half the chicks are disappearing every night, and no chicks are left after 4 or 5 days. The first opportunity we had to go back and repeat the study and therefore see if the 91 observations were typical was 2003, and essentially exactly the same picture emerged with a rather larger sample. So there's every indication that this heavy rat mortality of the pterodrome chicks is the norm on Henderson Island. And that raises the question of how the Henderson petrels, more or less endemic to Henderson, can survive at all. So I'm now going to just present a very simple model, and here are basically the calculations that we use, a breeding success that was observed, annual survival by analogy with other pterodroma species. These aren't data from Henderson petrels themselves. And then again, by analogy with other species, maybe a few breed at 4, most probably breed at 6, but even for older age groups, not every bird will breed every year. So you probably never get above 90% of the birds breeding in any one year. And we've got annual breeding, and you feed all those things together, and hey presto, you get a decline, but actually it's not particularly rapid. It's under 1% a year. So it's steady, it's relentless, but it's not going to finish the species in the next few years. Now let's consider whether the Henderson petrel could have withstood 800 years of this predation. So we know from the archaeological data that when the Polynesians arrived, they had petrels for breakfast, lunch and supper. So it's perfectly plausible that 800 years or so ago, the Henderson petrel population was maybe 10 to the 6, a million, conceivably even 10 to the 7th, 10 million. And then the two lines show the population decline that would occur if the rate of decline was that 0.82% a year that I showed you in the previous slide. And then we can insert today's population, and it in fact falls between those lines. It's about 16,000. So the point I'm making is that this heavy rat predation could have been going on for many centuries. We would still have a reasonable population of Henderson petrels, but if this scenario is at least roughly correct, the species is possibly going to disappear, not in our lifetimes, but in a century or two in the future. And then to avert that decline towards extinction, you can look to increase the breeding success, and you don't actually have to increase it very rapidly or very greatly, just 0.25 does the trick. Or you can increase adult survival, but there's no means that I know of that we can take steps to increase the adult survival of a species that covers huge tracts of sea feeding on small squid. And other tweaks to the little population model don't do the trick. So you can obviously see the direction in which I'm going. We're looking towards increasing the breeding success, and that means getting rid of the rats on Henderson. And that idea started to become seriously discussed in the early noughties, so in the years after 2000, and particularly after the success on the subantarctic New Zealand island of Campbell. That island of 112 square kilometres was cleared of rats in 2001, so at least in area terms, Henderson, Henderson, with 43 square kilometres, and this would be about half the island seen from the helicopter, is within the range of the possible. And of course the Henderson petrel would benefit from that eradication, but other seabirds that are more widespread nesting on Henderson would certainly also benefit, and doubtless there would be benefits to invertebrates and to the landbirds, of which there are 4 endemic species. So how would an eradication be carried out? Well, you would certainly need a ship to bring all the material, either from the west coast of the US or possibly from New Zealand, but as things came to pass, it was from the west coast of the US. You would need to drop the bait by helicopter, and the normal practice for these helicopter eradications is to have 2 bait drops, maybe 10 days or 2 weeks apart. And the plant phenology studies that we'd done during the main expedition of 91-92 indicated that maybe natural food was at a minimum in the months of August, September, at the tail end of the southern winter. However, when David Towns and I produced a feasibility report on this eradication possibility, we highlighted 2 issues that needed resolution. And the first concerns the crabs, and these are land-dwelling hermit crabs about the size of your fist that live really at quite high densities just behind the 3 main beaches, well, particularly behind the 3 main beaches on Henderson. And the problem associated with these crabs is that they eat the bait that, of course, you want rats to eat. They, as invertebrates, are not affected by the brodifacoon poison, but of course if they remove the bait, it isn't available for the rats. So you need to be sure that you put out enough bait to feed the crabs first, leaving enough over for the rats. So how you test this is by putting out dyed bait pellets without poison in areas where the crabs are most common. And what you are hoping to achieve is that a week after distribution of that bait, you will be able to come back and check the rats and find, all being well, that every rat shows evidence of having eaten bait. So maybe it has a pink bottom, maybe when you open it up, its guts are stained pink, or remarkably also, the whiskers grow out pink after consuming the rhodamine dyed bait. And we were able to tick that box. The other concern relates to the rails. As I mentioned, there are 4 endemic land birds on Henderson, a warbler, a fruit dove, and a small parrot, a lorikeet. Those 3 are probably not in any danger from the bait, but we know from New Zealand experience that rails certainly are in danger from poison bait pellets. And the Henderson rail is one of about 5 endemic rails left in the Pacific, and clearly it would be catastrophic to cause the extinction of a flightless endemic in the course of eradicating the rats. So there was never any argument that one needed to catch a group of rails, bring them into captivity during the bait drop, so that if the very worst happened and the wild population were extinguished, then the captive birds could be used to reseed, restart the wild population. So we established that we could catch rails, we could keep them in captivity, and so that box was ticked. And so we've now reached 2009, positive work from these studies, eradication was looking feasible. The approval from key parties like the Pitcairn Islands Council, Her Majesty's Government in UK, and the Invasive Alien Eradication Group, which is a group of mainly New Zealand experts who cast a wise and helpful eye over such projects. Then the RSPB in the UK took the lead in raising the money needed for this project, and planning proceeded for the project to happen in 2011. And the rail team on which I was arrived in early July, charged with bringing birds into captivity, and this plastic sheeting whose purpose we'll come to in a moment. And our target had been to bring 100 birds into captivity. For various reasons, we fell slightly short of that, but the total of 85 was thought to be adequate for the project to go ahead when the Aquila, the ship, the big boys arrived. And Aquila, it was in fact an Alaskan crab fishing vessel that had been chartered for this and indeed some other projects around the Pacific that involved island conservation. And this is the ship arriving on a rather dreary August day. And it might be interesting for people to see how these helicopter bait distribution projects work. We have the obviously the helicopter, and then a bucket that is slung under the helicopter. This contains about 300 kilos of bait, and then here is a motor which spins a rotor, and that throws the bait out to either side, about 40 meters either side of the helicopter's route across the island. And then the helicopter is flying on GPS traps that go zigzag across the island, and the various swathes of bait overlap, so that very literally no square meter of island escapes a bait pellet. And then, of course, it was vital when the helicopter was flying over the area where we had the captive rails that the rails be protected from the rain of death coming from the sky. And this we did by the very simple expedient of putting a plastic sheet, a polythene sheet, over the cages. And this actually worked well. Sometimes the pellets bounced off, and sometimes they were trapped on the polythene sheeting, but we had no rail casualties in the cages. So all was looking good up to the time of departure of the people looking after rails. The bait distribution, the bait drop, had happened in August, and the rail people left 3 months later in November. And then, alas, in March 2012, a National Geographic team were passing through the area. They were mainly doing underwater studies, but one of their team was onshore with a video camera, and he captured a short sequence of a rat, so there was absolutely no question of identification. There was a brief period of a few weeks when we could hope that for some reason this was the last surviving rat, clearly a very flimsy straw to cling on to, and indeed 6 weeks later in May, low-level rat presence was confirmed, and within 2 to 3 years the rat numbers were back at the starting level of 80,000 or thereabouts. So the project, I'm afraid to say, failed, and these projects of course are extremely brutal. You either achieve success and have a party, or you have to stand up and say the project has failed. And we could at least ask a few questions about the failure, and one question we wanted to know the answer to was, is the continuing presence of rats due to reintroduction rather than a failure of eradication? So very deliberately, samples had been taken from the rats before the eradication attempt, and then we could of course take them after the eradication failure and ask whether a reintroduction had happened accidentally. And of course the most likely source of a reintroduction was Pitcairn, and what this shows is obviously a fairly complicated tree, but I'll keep doing that when I don't mean to. What this shows is that the Pitcairn rats are shown in blue, and by and large they're distinct from the pre-eradication rats that were found on Henderson, which are shown in black. And what's completely evident is that the rats that were found on Henderson after the eradication, the red rats, cluster with the black rats, namely the ones that were on Henderson before the attempt. The red rats don't cluster with the blue Pitcairn rats. So we're confident that it wasn't a reintroduction, and all the information concerning boats, comings and goings, made that extremely unlikely. So the next question is, how many survivors were there? And we can use the change in allele frequency between the rats that were sampled before the attempt and the rats that were sampled after to get a handle on how many rats did survive. So the black dots show the modelled change in allele frequency according to the size of the bottleneck. So if the bottleneck is tiny, tiny, just single figures of surviving rats, then there's an abrupt change in frequency. The larger the bottleneck, so the smaller the change in allele frequency. The heavy black line shows the observed change, and the thinner black lines show the standard error. But what we're seeing here is that it looked as if the population, or the effective population size, was reduced to about 50 individuals, and that translates into a census population of about 80 survivors. So obviously these are very rough and ready figures, but it looked as if about 1 in 1,000 rats survived. But that, of course, is quite sufficient to generate a failure and huge disappointment. So what went wrong? As I've shown you, it seems not to have been a reintroduction. There were absolutely no sign of gaps in the helicopter coverage, no signs of batches of bait that were for some reason lacking the poison, Brodifacoum. Tests in 2013, so 2 years after the failure, showed no sign of resistance to the poison amongst the rats, which I guess were the grandchildren of the survivors. And of course you would expect those survivors to be the ones that were resistant, if any were resistant. So it does seem as if there were just a handful of individuals, 80 or so, that preferred natural food to the bait pellets. And follow-up fieldwork done in 2013 and 2015 actually confirmed the possibility that some rats may prefer natural food to those bait pellets, with the possibility that coconuts were an especial problem. And we had been alert to the coconut groves near the beaches, near the landing places on Henderson, and those had received extra heavy doses of bait from the helicopters. But a surprise that emerged from the helicopter flights was that there was a line of coconuts across the middle of the island, which we quite literally knew nothing about, because if you're cutting your path through the island, you can miss something that's 50 meters away. So the fact that we missed them wasn't a surprise. How they got there is frankly unknown. I mean, one possibility is that there was an American reconnaissance ship there on Henderson in 1966 with helicopters, and it's just conceivable that these coconuts were bombed into the island for fun. So where does that leave us for the future? Although the evidence in favour of an August-September baiting was not especially strong, there's no information that suggests any other season would be better. It would be nice if we could somehow select for rats that really like bait pellets, but I don't think that's practical. People are giving serious thought to how we might, in a future attempt, reduce the availability of coconuts. I think that would be difficult, but it certainly deserves more thought. The line I particularly plug is the possibility of developing a more attractive bait. The bait pellets are simply cereal pellets with poison inserted. Might there be the possibility of introducing attractants that overcome any reluctance of the rats to eat the bait pellets in preference to natural food? But as you've seen from the simple Henderson petrel population model, catastrophe is not imminent, and my feeling is strongly that one should wait 5 years, 10 years, 15 years until the possibility of success is demonstrably higher. And of course we have the New Zealand aspiration to rid that country of nasty rats and stoats by 2050. That project will undoubtedly develop new technologies that may serve to help Henderson and many other islands. I've now got just a few more minutes, and I'm totally changing track from the conservation aspects of this talk to talk about one of my favourite birds in the entire world, Murphy's petrel. And what's so enchanting about these birds is that they're nesting on the surface. It's supremely docile, and as I've explained when chatting to one or two people in the last week, you can literally just approach an incubating bird, take the egg out from underneath it, measure it, do the sort of thing biologists do to eggs, pop the bird back under the pop the egg back under the bird, and it seems totally unconcerned. And so it was the case that really on the back of the conservation work I've been telling you about, we've been able to do some tracking studies of Murphy's petrel, which for various practical logistic reasons is the most tractable of the four pterodrome species. And first off, I'm going to tell you about some GLS work, and this involves devices that were put on at the time of the bait drop in 2011, and then we retrieved the devices 2 years later in 2013. And we had considerable success, 25 devices deployed, 18 retrieved, so over 2 years we had 36 bird years of data. Tommy Clay takes all the credit for doing the analysis, and some of the findings were just wonderful in my view. So Murphy's petrels lay in, say, late May and June, so the southern winter, and then they completely leave the island during the southern summer, i.e. the northern winter. Where do they go in the northern winter? Well, they go to the North Pacific, and they're essentially in the central North Pacific, about on the latitude of the US-Canada border, and the immersion data from the loggers tell us that they're actually spending about 75% of the 24 hours sitting on the water. So basically their tactic is to go to the North Pacific and sit on the sea in the middle of winter. Why? I don't know, but it obviously works for them. And what this shows is that the two sexes, the males with, it's not very clear here, males with the diagonal shading and the females with the crosshatch are essentially using the same areas, very little difference. Also, I'm sure most of you are familiar with these chlorophyll A maps. Red equals the most productive regions, yellow less productive, green less, and blue absolutely hopeless. And we see, and we'll see the same in future slides, that the Murphy's petrel seems to be concentrating in areas of intermediate productivity. They're not going to the super productive areas, but they're avoiding the least productive areas. And then move forward to the pre-laying exodus. So this is the same map as we previously saw. This is the pre-laying exodus period, roughly speaking, April, early May. And this is the only time of year when there's clear sexual segregation. So the females are going around 1,000 kilometers south of Henderson. The males maybe 1,500 kilometers away. And again, they're going into waters that are more productive than those around Henderson, which is in the middle of the blue water desert, the South Pacific Jar. And this pattern of males going further than females seems to be one that is cropping up in quite a few territory of the species. And then we come to the incubation period, which is perhaps the most remarkable of all. And the next slide after this one will perhaps show things a little more clearly. But what we're seeing is really two areas of concentration, one to the south of Henderson, 1,500 kilometers or so away, and then another well away to South America, something like 3,500 kilometers away. And again, going to areas of intermediate productivity. And this is showing that, I think, a little more clearly. So Henderson here, the southern trips, showing the kernel density of usage on those trips, and then the long, long loops going out towards South America with a maximum distance, as I said, of around 3,500 kilometers from the island. And this, this map, and this is the brief plug for my book, which is very shortly out, but this map appears in that book, as do many other maps, as do some wonderful pictures from Bruce Pearson. And what I've very much tried to do in the book is really bring to a wide audience just the wonderful stuff that has emerged on seabirds thanks to modern electronics in the last 15 or 20 years. Many of you will have had a flyer in your conference pack. There are a few copies available for sale, so if you'd like to grab one from me at a particularly good price, make contact over the next few days. Going back to the science, universally these long trips, and we're talking with a sample of 20 or so, so this just shows 2 of the 20, universally the trips go anti-clockwise. So the birds initially head south, pick up the more westerly winds at 35, 40 degrees south, then reach their furthest point off Peru or maybe northern Chile, and then come back to Henderson at lower latitudes, maybe 15 south, benefiting from a following southeast trade wind. And then in 2015, it was really possible to follow up on those truly miraculous incubation journeys with some GPS tracking. And the GPS tracking showed exactly the same differentiation into the shorter trips to the south and the long looping trips away towards South America. But now I'm going to just focus on what happens in the first 24 hours after leaving. And I know this is small, so we're going to blow it up a little bit. And what we see here is that in the first 24 hours after departing Henderson, the bird, whether it's going eventually on a blue trip, a southern trip, or a long looping trip to the northeast, essentially goes off in the same direction for the same distance, mostly towards the southwest. There's no difference in departure difference for the 2 trip types. And then if we look at the pattern of landing, and the GPS birds also had immersion loggers, we see again And I think perhaps a little surprisingly that the first 24 hours after departure, maybe the first 48 hours, they scarcely land at all. And then only 2 or 3 days out do they start landing on the water fairly regularly. But again, you see no difference to speak of between the two trip types. And then if we look at where the birds land on the water during the entire trip, and I'm afraid it's not especially clear, but I think the message is reasonably clear. I mean, let's just focus on these long trips. Yes, there's the area in the first day or two when they're not landing, but then after that, the landings, as indicated by the blobs, are pretty much evenly spread throughout the trip. There's absolutely no sign of any obvious feeding destination. So perhaps they're feeding opportunistically for the entire duration of the trip after those first 24 hours. And to be perfectly frank, we're struggling to really nail down what are the advantages and disadvantages of the two trip types. And whilst of course we know that many petrels undertake two different trip types when they're feeding young, I'm not aware of any study that has shown two trip types when incubating. But at least the possibility comes to my mind that in fact they don't really know when they set out what sort of trip they're going to undertake. They head off southwest, and possibly if they find good food quickly to the south of Henderson, they embark on a short trip. If they don't find food quickly, they say, oh, bother, I'm going to have to do a long trip, and off they head to Peruvian waters. And then the final message from this slide is concerning the territorial waters involved. So on the short trips, so-called short, they're in fact outside Pitcairn territorial waters, they're outside French Polynesian waters. And that's even more true on the long trips. Not only are they outside French and Pitcairn waters, they're largely outside Chilean waters, either from Easter Island or from the islands closer to the coast, and they never even enter Peruvian waters. So I think the message there is that most landings are in international waters, and maybe Murphy's petrels are in a group of species for which any sort of marine protected area approach is frankly problematical at best, and may be just a waste of time to even think about. So let me really sum up. In terms of the future for Henderson, I think the island will survive. It's secure because it's so remote, and therefore the best thing is to wait and before undertaking a second attempt, wait until we can be as confident as possible that that second attempt will be successful and get rid of the rats. And there are a certain group of seabirds where at-sea conservation may just be problematical forever, and maybe we have to accept that, and of course make conditions onshore as favourable as possible for such species. So that brings me to the end, and thank you very much for your attention. Thank you.